Okay, folks, so in part 1 of chapter 13, we talked about how viruses are different from bacteria or living cells. We talked about the components of viruses. We talked about viral replication. And we ended with a note that viral replication is going to start based on the genetic information that that virus comes in with. So let's kind of pick up there. So we said that viruses come into their host, or viruses have genetic information that is either DNA or RNA, never both. And that genetic information is either double-stranded or single-stranded and not both. And so if you think about how the host cell is replicating its DNA, it's going from DNA to RNA to protein, then you can imagine that how the virus comes in with its genetic information is literally just going to engage within that process, within that step of the central dogma. So what does that mean? Well, say a virus comes in with double-stranded DNA. Then those viruses are going to actually migrate to the nucleus of the cell if it's a eukaryote, or to wherever the DNA is aggregated if it's a prokaryote, and it's going to engage at the beginning steps of that dogma. It's going to look like the DNA of the cell, and then it's going to be transcribed and translated into its various components and proteins, right? Whereas if a virus comes in with RNA, then it's going to mimic some sort of RNA, mRNA, or precursor to mRNA, and it's going to jump in at the step right before translation, right? And it's going to engage with the ribosomes and go through and produce the viral proteins and the components that need to be made. And so, depending on how that virus comes in, if it's double-stranded, it's more likely to start off double-stranded DNA, start off mimicking the host DNA, and it's going to go through all those steps of transcription, translation, so that it can make its viral components. Whereas if it comes in as RNA, so maybe it comes in as single-stranded RNA that's positive sense. Well, what does that mean? Positive sense means it looks just like an mRNA molecule and it's ready to feed right through a ribosome. Maybe it comes in as negative sense. Negative sense means that it's the complement of what the mRNA molecule would be, and it just needs to be, converted into its complement, and then it can run through just like an mRNA right through the ribosome, right? Maybe it comes in as double-stranded RNA, where those two strands can be separated, and one can be a template for mRNA molecules, and one can act just like an mRNA molecule and run through the ribosome. Okay, and then we have some really unique RNA viruses called retroviruses that actually come in with an RNA template, backtrack using an enzyme called reverse transcriptase, and make DNA. The DNA goes in and becomes a prophage in a latent or, a cycle, latent stage within the DNA. So it hangs out in the DNA through a lysogenic cycle, but it's actually sort of active, which is oftentimes prophages are not active, but in retroviruses like HIV/AIDS and some of the other, viruses in this particular group, then they are actually backtracking from RNA to DNA. So that's a really unique group of viruses. Really unique group of viruses, but they are RNA viruses. And so, regardless of the virus, they're always going to jump into the step that's appropriate based on the genetic information that they come in. So if it's double-stranded RNA, you can see here those double strands are coming apart and becoming a template, and then also an actual mRNA and a template to mRNA. You can have your single-stranded negative sense that's a template for mRNA, or you can have your single-stranded positive sense, which just looks straight like an RNA virus, an mRNA molecule. What's not in this image here is the double-stranded DNA, which would come in looking like the double-stranded DNA of the host, and then just go through all of the steps of transcription and translation. But in that way, we can see that based on the genetic information that the virus comes in with, it will essentially hijack different steps within the central dogma or the process of, a protein production that the cell or the host is engaging in. Okay. So that is basically what we'll cover for viruses. There are two sections, though, that I would recommend that you read. So there's a section on the role of viruses in cancers, because there's a significant association and even a causative association between viruses and many human cancers, which makes a lot of sense because the virus essentially is hijacking the host, removing its ability, the host's ability to engage in its normal biological functions, and up regulating the replicatory or the replication machinery of that host. So what you end up with is a host cell that's not functional but is still replicating. And that definitely will lead to benign or malignant tumors, or potentially will lead to benign or malignant tumors if it's not stopped. And we'll talk about within our immune system how we actually have components of our immune system to look for such said cells and mitigate the risk. Okay. There is also a section on culturing viruses. We already talked about within the lab, we cannot grow or culture viruses on their own. We either need to culture them on like live embryos or live bacteria, or even cell cultures like human cell cultures. And so definitely read those sections so you have an idea about how culturing looks like in the lab. Relevant to both of these sections, there is a book that's written by a former PCC student that you may have heard of called The Immortal Life of Henrietta Lacks. And this gets into a lot of the intrinsic kind of institutional injustice and racism that comes about when we're studying and how we know what we know about viruses and various types of cancers and so on and so forth. And so that is a really important and relevant book to both of these sections. So I recommend that you check that out. Okay. So the last note that we want to make before we end and conclude our section on viruses is something that we've already discussed. Are viruses alive? So what we've seen in this lecture, or seen in this chapter, is that viruses, they come in with machinery that mimics the cell, right? They have genetic information, they have a couple of proteins, and they have the tools to engage with the actual machinery of the cell, but they themselves do not have all of the components that they need to actually carry out metabolic processes, reproduction, response to stimuli. They don't have functional membranes, for example. And so they're really interesting because they teach us about the basic components of life, even though we don't consider viruses alive in and of themselves. They also remind us that within biology, we like to, and science generally, we like to categorize things as living or non-living. Maybe if we go back to our discussion of prokaryotes, gram-positive, gram-negative, but there are always exceptions, and these exceptions help us understand these kind of principles or these groupings that we might have come to based on the research that we have. And so it's really interesting to kind of think about how viruses teach us about living systems, even though they themselves are not alive. Okay, so that is the first section on viruses for chapter 13. Go ahead and pause here, review your notes, and the discussion post for chapter 13 is that I want you to go and look at the virus SARS-CoV-2, which is the causative agent of COVID-19, and tell me about its characteristics, the characteristics that we talked about. What is its genetic makeup? What does its capsid look like? What types of proteins does it have? Is it enveloped or non-enveloped, right? And give me some information about SARS-CoV-2, the causative agent of COVID-19. And so that's going to be our discussion post for chapter 13. Once you come back into the lecture, there's still a little bit more, come back into the lecture, we're going to talk about prions and then we'll conclude our discussion of chapter 13. Okay, see you in a sec. Okay, welcome back, folks. So the other discussion that we want to have in chapter 13 is of prions. And prions are fascinating because they are also acellular agents that can cause disease, but they're much, much simpler than viruses. So with a virus, we have this genetic material that's organized within capsid proteins that are protecting it. Maybe there's an envelope even more, above the capsid of that virus. So it does look like an organized component with similarities to living systems. But prions, on the other hand, are just molecular components that are generating disease, right? And so what a prion is a protein, just like a protein that our body makes, that's misfolded. And that misfolded protein actually creates a domino effect that leads to the misfolding of other adjacent proteins. And those other adjacent proteins that are misfolded, as they aggregate, they make these non-functional plaques that then are eliminated by the body. And this is what generates disease. This is what actually degrades the brain and its functions. And all prion diseases that we know of, all prion diseases are eventually fatal. Okay. And so let's talk a little bit about prions. Okay. So there's this protein in the brain called cellular PRP. It's in all mammalian brains, and it's important to signaling and, through via neurons and synapses and so on and so forth. Some of the functions we know, some of them we don't know. What happens is the polypeptide chain for the PRP protein should be folded in a very specific way. Okay, so I'm going to show you an image of that. Here's a normal PRP protein. And that's a stable form, and that's the functional form, and that's what most of us have in the cells of our brain. Now, there could be an environmental factor like a prion that's ingested, or a genetic factor, so a mutation in this in the gene for this protein, or another environmental factor that leads to the misfolding of this protein. Now, when I say misfolding, what I actually mean is that there's another stable folded structure for the same polypeptide. Okay, so we know that we have hydrophilic and hydrophobic regions. We have different types of beta and various types of sheets that are in there, and they fold in on each other based on their molecular interactions. And so you have this stable form that is normal. But there is another stable form that is abnormal and mutant, but it's still molecularly stable, right? And I actually have a 3D model here thanks to a student who did their term project and artwork on prions. So here you have the normal folded structure of your prion, okay? And that's right here. You can see that normal folded structure. But then you have this abnormal folded structure. It's the same polypeptide, but it's folded in a slightly different way. And that's over here in this image. This is the abnormal folding of this structure. And this abnormal mutant prion is not functional, but not only is it not functional, every time this abnormal prion runs into a functional PRP protein like that, it will actually misfold it. And then that misfolded protein will run into a normally folded protein and it'll misfold it. And then you get these aggregate of misfolded PRP proteins that are mutant that create plaques in the brain because they aggregate together. And then your body actually identifies them and eliminates them. Okay? And so this is why The diseases, prion diseases, oftentimes are called spongiform encephalopathies because they actually, as your body eliminates these plaques, they actually make holes in the brain tissue. So you can see that here. Here's the normal brain tissue. Here's a spongiform encephalopathy, where you actually have literal holes or sponge-like structures within the brain as the brain is eliminating those plaques. So it's very interesting that a protein can actually be a disease-causing agent. Okay. And so we're going to watch a couple of videos that give us an idea of how prions work and what types of diseases they cause. So let's start with just kind of how these prions work molecularly, and then we'll watch a video about some of the diseases that are caused. Okay, here we go.
All mammalian cells contain a gene that codes.
1 second, let me switch the audio here.
Primary sequence of amino acids for the prion protein PRP. Normally, PRP folds into a functional form with several alpha helices called cellular. The normal.
Okay, I'm going to start that over just once more. So that we can hear better.
All mammalian cells contain a gene that codes for the primary sequence of amino acids for the prion protein PRP. Normally, PRP folds into a functional form with several alpha helices called cellular PRP. The normal function of the prion protein is not well understood, but it's believed to be important in normal synaptic development and function, and it may be involved in stabilizing the structure of the synapses and in establishing memory. The prion protein is also capable of folding into a form with beta-pleated sheets. Prions do not reproduce like bacteria or viruses. Instead, infectious prions convert normal prions into more infectious prions by folding them into beta-pleated sheets. Thus, the number of infectious prions is increased, but by conversion rather than traditional reproduction. Prion proteins in the infectious conformation can group into multimers, which are very stable and resistant to protease. Over time, these multimers probably lead to some of the damage seen in prion-infected tissues.
Okay. And here is another video that goes over some of the diseases caused by prions. But you will see that you can actually kind of see the flow of prions even through the food system, for example, as they're very stable molecules. Here we go.
There are several human diseases known to be caused by prions. Creutzfeldt-Jakob disease is rare, occurring in about 1 out of 1 million people. It usually appears in midlife between ages 20 and 70. The average age of onset is about 50. Recently, a form of the disease called variant CJD, VCJD, has emerged. It was first detected in the United Kingdom in people who had eaten beef from cattle infected with bovine spongiform encephalopathy, so-called mad cow disease. Once the first symptoms are seen, both variants of CJD progress very rapidly from onset to disability to death. CJD affects the cerebrum, where it causes holes called vacuoles and amyloid plaques, clusters of prion proteins and infected cells. Gerstmann-Straussler-Schenker syndrome, GSSS, is a rare human prion disease. GSSS is caused by a single mutation in the gene encoding the prion protein and is characterized by amyloid plaques in the brain in several areas, mostly in the cerebellum. The disease causes eye-body coordination difficulties, intellectual disturbances, and eventually dementia and death. Fatal familial insomnia is a genetic disease caused by a mutation in the gene encoding the prion protein. The onset of the disease usually occurs around age 50. The structure in the brain that is most affected is the thalamus, which is responsible for the regulation of circadian rhythms. The first symptom of fatal familial insomnia is a progressive insomnia, which eventually leads to complete lack of sleep and coma and death a year and a half after the onset of symptoms. Kuru was a prion disease found among the South Four tribes of New Guinea starting around 1900. Kuru was also known as laughing disease because the victims would have uncontrollable laughing fits as their symptoms progressed. Kuru was found to be transmitted by brain matter of diseased relatives, which was handled and eaten during mourning rituals. When cannibalism was outlawed in the region in the late 1950s, the disease sharply declined, and cases of Kuru have since vanished. Kuru mostly affected the cerebellum, and the most pronounced symptoms were related to motor skills. Several animal diseases are also known to be caused by prions. Scrapie is a fatal degenerative disease that affects the central nervous system of sheep and goats. Scrapie has been known since the 18th century, and it's believed that bovine spongiform encephalopathy was transmitted to cattle through the practice of using contaminated sheep bone meal in cattle feed. Infected sheep often scrape themselves against objects until their skin is raw, giving the disease its name. Eventually, the infected sheep lose motor control and die. Transmissible mink encephalopathy, TME, affects the central nervous system of ranch-raised mink. TME has an average incubation period of more than 7 months before the onset of symptoms. Symptoms can last from 3 days to 6 weeks. Early symptoms include an increase in nest soiling and spreading of droppings in the cage. Microscopic examination of the brains of infected minks shows that the disease is limited to the central nervous system, causing sponge-like changes in specific areas of the brain. Epidemiologic studies suggest that animals contract the disease by external exposure to the infectious agent, such as by eating contaminated feed. Chronic wasting disease is a fatal neurological disease found in deer and elk, caused by prions. The disease attacks the brains of infected deer and elk, causing the animals to become emaciated, display abnormal behavior, lose bodily functions, and die. This disease has been found in both wild and captive deer and elk. Bovine spongiform encephalopathy, BSE, is the so-called mad cow disease that brought prion diseases to popular attention. BSE is a transmissible neurodegenerative fatal brain disease of cattle. The disease has a long incubation period of 4 to 5 years. BSE is transmitted through the consumption of BSE-contaminated meat and bone meal supplements in cattle feed. Affected cattle behave erratically and begin to lose motor control and eventually become unable to stand up. Infected cattle are sometimes referred to as downer cattle. It's believed that some humans acquire the variant Creutzfeldt-Jakob disease from eating contaminated meat from downer cattle.
Okay, so that gives you an idea of some of the different prion diseases that we know of in mammals. Okay, so prion diseases are pretty rare, but still there are some similarities between some of the misfolding of proteins that happen with prion diseases and some of the impacts that we see in Alzheimer's, Parkinson's, ALS, which have some, less likely to be, 100% fatal like a prion disease is, but still has some kind of similarities in terms of the way that proteins are misfolding potentially in the brain and causing some of these degenerative symptoms. And so it is still important to be able to understand prions for prion diseases, but also for their implications potentially in other diseases as well. So that is prions for us. Even though prions are just a protein, they are still potentially a disease-causing agent, and they give us an understanding of how that function of proteins is associated with their structure. And when you change the structure, you could potentially, and often you will, lose the function of that protein. And so that brings us basically to an end of chapter 13, where we've learned about how viruses and prions are acellular agents that could be disease causing oftentimes, that are different from bacteria, and living cellular components that have all of the characteristics of life, but viruses and prions are able to impact living systems and cause disease. And so that's essentially chapter 13. Let me know if you have any questions. Go ahead and review your notes before you close out, and make sure to do your discussion posts on SARS-CoV-2, which is the causative agent of COVID-19. Thanks, folks, and I'll see you in the next lecture.